Hello, everybody. I'm David Pellman, and it's a tremendous pleasure to be able to introduce Gowden's Dan User, who's going to give one of our special oncology seminars, one of the small number of endowed lectureships. So Gowden's is going to be, this seminar is funded by the Weiner-Levin family and was established in 1997 by the late Rose Weiner-Levin and her children. The purpose of the lecture is to further research within human cancer genetics. The Levin family remains committed to accelerating improvements in healthcare for patients and their families, and we're very grateful to the Weiner-Levin family for their long history of supporting Dana-Farber's dual mission of cancer research and cancer patient care. So our lecturer today is Gowden's Dan User, who has a long and somewhat peripatetic path. Gowden's is currently the Patrick Haggerty Distinguished Chair in Basic Biomedical Science and the Secret Scholar for Cancer Research at the University of Texas Southwestern. He's also the inaugural chairman of the Lydia Hill Department of Bioinformatics, and he's the director of the Cecil and Ida Green Center for Systems Biology. Gowden's started out, his career developed out of a mix of music and mathematics. He was an undergraduate at the Music Academy of Bern, where he trained as both a composer and conductor, and where he won awards for his composition. And then he took a slight shift into electrical engineering and got his PhD in the ETH in Zurich. And that was followed by a postdoctoral fellowship with the famous Shinya Inoue at Woods Hole. He then migrated to the Scripps Institute in San Diego, where in the span of 6 years, he went from assistant to full professor. And then he moved again, and where I had the good fortune to meet him, where he came to Harvard Medical School, where he was at from 2009 to 2014, before UT Southwestern lured him away. I have tremendous admiration for Gowden's work and have so for many years. He brings exceptional rigor and deep biological understanding to imaging what goes on in cells and thinks deeply about the broader biological significance. And today he's going to teach us about cell morphological control over oncogenic signals. Thanks so much, Gowden, for sharing your talk with us.
Thank you, David. Thanks for the invitation. Thanks, everyone who I cannot see for being here and listening to what we have to share with you. And also would like to thank the Weiner family for this wonderful opportunity to speak at the Dana-Farber. So, as David said, what I'm going to ask today and try to give you some answers is how much does actually the shape of a cell control signals, and in particular in the context of cancer, control oncogenic signals. And the enabling technology by which we can begin to ask these questions is, as shown in this first slide, light sheet microscopy. So with my move from Harvard to Dallas, we really undertook a major effort in developing light microscopy that is fully 3D. So cells are sitting in more and more native environments, here just shown in collagen gels, without doing any compromise on the resolution of the cellular and subcellular features we want to image. And this is really a dual effort. On the one hand, you need to build specialized microscope hardware, which is done by Rita Fjolk and Kevin Dean. You need also on the biological side, really the right preparation of the samples. And this was spearheaded in the beginning by Eric Welf. But it is also, as you certainly appreciate on this slide, an enormous computer science challenge, which is my background, as you have heard, and Megan Driscoll was the first postdoc here in Dallas who really took on that challenge. And to show you how complex this is, I have here a first C-slice sequence that goes through that very data stack you see here on the left side, where we have to segment out cells. And then what you see here color-coded on these cells is actually the curvature, the local curvature on that dendritic cell surface. So the resolution as well as the accuracy with which we can reconstruct these individual cells is so high that we can actually begin to play mathematical games like differential geometry. And that is very important to keep that in mind in the background of the biology stories I'm gonna tell you. I will not belabor too much the technological developments that are necessary to get at the biology any further. So with this technology in hand, one of the earliest observations we made, this is a melanoma cell here, which sits again in one of these collagen gels, which we don't show here now, is that you have these morphological behaviors like blebbing in this case, which seem to actually attract signaling activities. So what you see here in colors is not the curvature, but it is the result of a biosensor which monitors the activity of PI3 kinase activity. And I point out here in the lower left corner, it is really the morphology which seemed to happen first before the signal comes into play. And this is not completely surprising when you go back to the biophysical phenomena. When you squeeze a round cell to become flat, and that is what we do quite often in cell biology when we culture our cells and tissues, you naturally upregulate signaling activity just by bringing molecules into more proximity, in particular also into higher proximity to the receptors, which are at the cell surface. And there are other morphological cues that have sort of the same effect. You have narrow spaces in cells can act as diffusion traps, or blebs like the ones you have seen before can lead to aggregation of receptors, or aggregation of curvature-sensitive activators of signals, or to scaffolds. And as these morphological programs ensue, you also have mechanical activation. And as we all know, there are a lot of receptors, such as integrins and others, which are directly force responsive. But where things really get complicated is now that all these and probably many more morphologically responsive cues are really combined and lead to potentially very localized feedback interactions that really might upregulate or downregulate certain cellular signals, as you have seen in the example of PI3 kinase. So before we go into details of that, I would like to sort of have two slides which bring us into a general question. So we often think in cell biology downstream of the central dogma about signaling. And then if you're interested in cell morphology, you think about cytoskeleton architecture, which controls morphology. And these transfers from signal to cytoskeleton morphology, we all understand very well, are driven by the environment. Dimensionality of the environment matters, mechanics matters, and the ligation also of the environment can lead to signaling. That's why it's so important that we build technology that allows us to look at these processes in environmentally as least limiting conditions as possible. But for this afternoon, the key question is now, is potentially this morphology that is at the bottom of the whole pathway feeding back into signals? And in the context of cancer, are these signals driving hallmark functions of cancer cells, such as proliferation or survival? And so for that, I would like to stimulate now a discussion through a little bit of a thought experiment. It's actually a fun experiment we really did in the lab, which can be entitled by finding an unbiased evidence for morphological control over, in this specific case, metastatic cancer cell function. And in 2022, and as a computer scientist and chair of a bioinformatics department, I have to quickly talk about deep learning. So it's going to be a deep learning story where really AI will test whether such evidence is here. So the beginning of the story is a collaboration with my colleague Sean Morrison here at UT Southwestern. Sean has over many years developed a melanoma, a PDX transition program where he cultures these cells in melanoma, passages them to the next mouse, and so forth. And in this particular paper in 2012, he showed that he can really maintain correlations between the melanoma in the mouse and what happened in patients. So very specifically, all the samples we got now from this mouse passaging assay are stage 3 melanoma. Most of these melanoma metastasized to distant organs in mice, but the fraction only metastasizes to the lungs. And the subsets that did not efficiently metastasize are actually originating, and he has a lot of evidence in this particular paper. They originate from patients that did not die from metastatic melanoma. So we had a population of PDX tumors which are highly or less highly metastatic. And what we did is we just stuck them under the microscope, filmed them for 4 hours with a phase contrast imaging setup. And I don't even know anymore which one is high and which one is low in metastatic propensity. You can zoom in and look at the individual cells, and they're dancing around. And so here comes now the AI. What we did with AI now is ask a computer to give us 56 numbers that are extracted from these movies, such that given the computer again presented with these same 56 numbers, can reconstruct the same movie as best as possible. So in other words, what we ask the computer to do is extract 56 numbers which represent a cell in the best way. And this deep learning network, which is symbolized by this encoder, so you encode features and then you decode these features again back into images. These deep learning networks are trained not only on melanoma, but on a more general class of cells. And there are a couple tricks which I don't want to belabor. So you can look here in this movie at how this training happens. This is the input, and these are the reconstructed images, which are automatically learned by, again, extracting 56 numbers. So when we take now these 56 numbers and pump them into a classic machine learning classifier, then we have very, very high accuracy in distinguishing high from low metastatic tumors. When we do the same thing at the single cell level, as shown here by these distributions, you have, of course, in low metastatic tumors, also cells which go into the high efficiency arena, which is here between 0 and 0.5, and vice versa. And that's not a surprise, because we are really dealing with the heterogeneity of cancer cells in this case. But at the population level, we have very precise prediction. And so how good is this prediction in reality? So what we do now here is we took a panel of melanoma cell lines, so not PDXs, and classified them with the same trained system we used to classify PDXs, and found 2 particular cell lines, A375. The melanoma people on this call certainly are familiar. This is a garden variety melanoma cell line. And MV3, A375 as the highest metastatic, and MV3 as the lowest metastatic population of cells. And when we stick them now into mice and use a spontaneous metastasis assay and read out after about 30 days, where do these cells metastasize, if at all, then you see very clearly in a bioluminescence image that the A375s metastasized into many different organs, while MV3s do not metastasize, largely do not metastasize. Very interestingly, at the level of the primary tumor, there is no difference, or if there is any actually, then it is the more metastatic form, A375s, which grows slower. So in other words, what this computer system has figured out is these particular features which are really predicting metastatic propensity. And that is now, from a computer science point of view, the interesting question: what is this system learning, right? And so here we played a nice mathematical trick, which I'm not going to go into the details of. I'm happy to explain that later. Is what we do here is we take now these 56 numbers, which are in a so-called latent feature space, and check which of these numbers correlates best with the final classifier score that distinguishes low from high metastatic cells. And so in this particular case, we had one outstanding feature, but it does not necessarily need to be a single feature. It also works for a combination of the features. But here in this specific case, we had one outstanding feature, feature 56, which highly correlated with the confidence score for high versus low metastatic propensity. And so once you have that, then you can actually play now a wonderful game, since this is going through these autoencoder systems where we encode 56 numbers and use these 56 numbers again to generate images. We can now go into that space and say, okay, let's push feature 56 for a melanoma that is classified as inefficient into the efficient metastasis space. And we can do that not only within the variation, the natural variation of our data, we can actually extrapolate and generate now movies which do exactly that. So make an inefficient metastasizer efficient. And what you see really as the outstanding features is these cells begin to have, in a synthetic fashion, pseudopodial extensions. And you can now go in the other direction. And so this movie loops here. We take an efficient metastasizer, which has these pseudopodial extensions, and we make it inefficient by moving it to the right side in the feature space. And essentially, you take these extensions away. There's also a little bit of a side story going on in light scattering, but there is definitely a morphological feature that seems to be coupled to the metastatic propensity. And so we were not really surprised by that result, because we had actually 2 years earlier published a paper in Developmental Cell, which really established these killer lamella in melanoma as being morphological programs that drive drug resistance as well as metastatic proliferation. And so I'm going to tell you a little bit about this story, which was really spearheaded by a fantastic MD PhD student, Abby Mohan, in the lab. And the starting point for these lamellipodia is actually a very peculiar mutation in the GTPase RAC. RAC is really very well known as a driver of cytoskeleton activity, and in particular protrusive activities of cells. A little more background on this RAC. It is the third most frequent mutation after BRAF and NRAS in melanoma, and it was identified concurrently in 2 labs, one at MD Anderson, Linda Chin's lab, who many of you might be very familiar with, as well as in Ruth Balaban's lab at Yale, published both in 2012. And so what this mutation is, it's a C2T variation in codon 85, which leads to a prolin to serine mutation in the switched on domain of that signaling molecule. And essentially that leads to a release of a conformational restraint. And with that, a faster GEF activation when the signal is activated by an upstream GEF. So what is very important, this is not a constitutively active signal like the RAS mutations or the RAF mutations we are all very familiar with in the cancer world, but it is a mutation that is still completely regulated by upstream components. But once it is active, it is a little more active. And Linda Chin's lab then 2 years later, after the initial publications, showed that when these mutations are expressed in BRAF mutated cancers, these cells do respond much less to directly BRAF inhibitors as well as MEK inhibitors and MAP kinase inhibitors. So red is the P29S line compared to those response curves of RAC and GFP expressing cells. So we took that result and reproduced it in our A375 cell lines, which express the P29S cell. On the DMSO, there is absolutely no difference between a wild type and the P29S mutated RAC expression. However, when you go and challenge now these cells with, again, the brafenib as a BRAF inhibitor, as well as trametinib, which is a MEK inhibitor, then you see that the P29S cells have a massively higher proliferation and sustainability compared to the wild type cells. And we can also you do the reverse experiment. We took a cell line which naturally expresses the P29S mutation, reverted it back through CRISPR into wild type, and see essentially the same phenomenon. P29S cells under drug challenges have a much higher proliferative potential than the reverted wild type cells. When we stick these cells into mice again and use spontaneous metastasis assays, then you see very strong upregulated metastases in the case of P29S expressing melanoma cells. And when we look now in particular in the lung, then you see really beautiful, or not so beautiful, but very strong metastatic nodules. And so with P29S, of course, there's two hypotheses. Is P29S this RAC mutation that drives cytoskeleton activity, just increasing metastasis through migration, or is it really truly also in the case of metastasis a proliferative response? And so we tested, or Abby tested that by imaging in histological sections through phosphohistone 3 marker the proliferation potential, and as with the drug challenge, it is really P29S driving primarily proliferation. So we think that while we cannot exclude that there is a migratory effect as well, proliferation certainly is at play. And very much like in the case of the AI system, which can classify metastatic potential, also in this case here, there is no advantage of the P29S expressing cells in growing primary tumors. If anything at all, again, it's actually even slower. So that brought us to the hypothesis that this mutation really protects proliferation in melanoma cells selectively when they are stressed either by drug challenges or when they have to colonize foreign tissues. And so how does this happen? Well, we have here the canonical MAP kinase signaling pathway. We go into cyclin D1 and proliferation, and then obviously what dabrafenib and trametinib do, a drug is block that upstream input to ERK signaling. And when you now look again with the P29S cells, there is a slight shade of still phospho-ERK, so active ERK going on. But what is very striking is that compared to the wild type conditions, the P29S condition really has highly upregulated cyclin D1 activity. So we were wondering whether there is a leakage of P29S into ERK, and there is a number of pathways which are known between RAC and ERK activation that would be sufficient to lead to that slight upregulation phosphorylation, and that in itself would then be again sufficient to drive the cyclin D1 response. Well, we tested this by going directly with ERK inhibitors into the same system, where we can now completely eliminate phosphorylation of ERK, but cyclin D1 is still equally upregulated in its concentration. So that meant that there is some form of a direct pathway between RAC P29S and cyclin D1, which is not activated when you have a RAC wild type situation. So how does that work? And that was 2 years of really dancing around in the dark until we really carefully looked again at images, right? And so Abby came the other day into the office and showed me these pictures where she said, okay, here's a wild type RAC expressing melanoma, and when we pour drug like dabrafenib on these cells, then they become spindly. And this has actually been described by many other labs working with melanoma. This is a very well-known phenotype. However, when we look at the P29S cells, they're already, with no drug, a little more puffy. But when we now go with drug and challenge these cells, then they massively get upregulated lamalipodia and look really like fat fried eggs, right? And so we can image this now live, and you have seen that movie already. What really happens is that under this drug challenge, these cells develop this wide lamalipodia. I've studied lamalipodia for 20 years. I've never seen in my life such a wide lamalipodia. And as computer scientists, of course, we developed in software to really rigorously quantify that. And you see very clearly that while wild type has a slight upregulation of lamalipodia 2 under drug challenge, it is really on the P29S mutations that you have this strong, very strong morphological response. So that brought us to the question, are these enhanced lamalipodia really the drivers of MAP kinase inhibitor insensitivity of these cells? And that's an experimental challenge to test. We now somehow need to manipulate MAP kinase inhibitor sensitivity through lamalipodia. Not easy at all, because you immediately get wholesale effects if you begin to play with morphology, right? And so how we did that, for that I need to give you very quickly some background on the details of Lamellipodia. Lamellipodia form through the assembly of actin filaments, where you have mother filaments, most likely nucleated through members of the formin family and others, so linear filament growth. And then as the forces begin to kick in, as the filaments begin to push against the membrane, you have now a feedback into RAC signaling, which now upregulates a dendritic filament growth through this ARP23 actin-related protein complex 2,3, which has the ability to branch off new filaments from pre-existing filaments, and through that goes into exponential growth, right? So it's a very strong reinforcement that happens on that particular nucleated system. And so the hypothesis now was that with P29S, which can be just upregulated, normal RAC activity a bit more upregulated, that we just would enhance that feedback loop and therefore drive that ARP23 response. And so what we did then is took now ARP23 inhibitors. There is a nice inhibitor out there, CK666, where on top of the dabrafenib treatment, we titrate in more and more ARP23 inhibition, right? And so you see here that while without CK666, we have this immediate response of wild type cells to dabrafenib. You have seen that data already, as well as with CK666, the wild type cells do not care. The P29S cells very much care. And if you could pour more CK666 without massive perturbation of many other cell functions, you would probably be able to bring this down to the same levels as the wild type. Very importantly, as a control experiment, when you go now with pan-formin inhibitors, which just block that linear filament growth, then you don't see that effect. So in other words, for this P29S effect to happen, you really need a dendritic actin network. So there's two hypotheses. Either you just need a dendritic actin network, or as you grow a very strong network, you generate forces, and then in sort of a Val-Weaver type response, you would now upregulate integrin mediated pro-survival and pro-proliferative signaling. We did a lot of tests and came to the conclusion that this is not the case. However, peculiarly, you still need some form of adhesion. You don't need integrin mediated adhesion necessarily, but some form of adhesion is necessary so that the cell can keep these very flat structures, right? While if they lose adhesion completely, they will just ball up and become round. Okay, so what is going on, right? What is going on here is that this Lamellipodia, which is a very thin flat structure, typical about 200 to 500 nanometer thin, is pumped full with these dendritic actin filaments. And that sequesters now a tumor suppressor, NF2 Merlin, which has actually actin binding sites. And so essentially takes it away from the cytosolic and the nuclear spaces so that it can no longer act as a tumor suppressor. And on top of all that, RAC as a downstream effector has PAC, which is a kinase that phosphorylates NF2 directly. And through phosphorylation, you inactivate biochemically further the action of this tumor suppressor. So there is a double effect here. And so this was really the first example we think we had, which had really a morphological program directly upstream of an oncogenic signaling cascade that was now controlled. And so with that, we come to the first summary. So Lamellipodia in this particular case do affect signaling, and in this specific case leads to the NF2 deactivation, which enables proliferation. So I would like to come back to that PI3 kinase blebbing movie, right? And that's the second story I'm going to talk about. This is an unpublished story, really driven by Andrew Weems, who I think is in the audience on the other side of the Zoom wall. And he was assisted again by computer scientists, Megan Driscoll and Hanye Maslum Farsibaf. All this type of experimentation really needs a lot of very original data analysis, which I'm really not talking about this afternoon. So, BLEBs. Well, very importantly, the blebs I'm going to talk about are not apoptotic blebs, but these are dynamic blebs. And they form over the time scale of about 2 to 3 minutes and have really nothing to do with apoptosis. On the contrary, as you will hear, these blebs actually make cells highly happy and surviving. So what is a bleb? A bleb is understood as a program that starts with a detachment of the plasma membrane from the actin cortex. And since intercellular pressure is a little higher than the extracellular pressure, then that opens up the opportunity for the plasma membrane to unfold in a balloon, which gets bigger and bigger. And then at some point, the actin cortex begins to assemble inside the bleb. And as soon as myosin, so an actin-associated motor comes in, it begins to contract and the bleb withdraws. And as I already mentioned, this happens over a time scale of 2 to 3 minutes, right? So you can take a garden variety of non-transformed cells like MEFs or HEX cells or epithelial cells. And when you detach them from the substrate, these cells begin to build these dynamic blebbing programs, as illustrated here in these insets. Right? Well, for a normal cell, what happens here in the case of a normal fibroblast, 1 to 2 hours after detachment, these cells can no longer bleb. And that is because they probably have used up all the excess plasma membrane they gain when they get detached. They have a lot of excess membrane, and that is potentially endocytosed. And without reattachment to the substrate, they undergo a programmed cell death, which is known as anoikis. While melanoma cells, they have this ability to indefinitely bleb, and therefore are anoikis resistant. And I'm pretty sure the majority in the audience is very familiar with anoikis, and anoikis resistance is really a hallmark of metastasis. These cells need, as they crawl out and invade into tissues, in particular when they get into the bloodstream, they need to be able to deal with attachment-free conditions where they do no longer receive signals from the neighboring cells or the substrate. And indeed, blebbing is a very well-described phenomenon at the front of invading tumors, here beautifully shown by this work from Eric Sahai's lab. Through imaging, melanoma cells do start to bleb and go into this so-called amoeboid morphotype when they begin to invade spaces proximal to the primary tumor. But these blebs have really been associated in the literature mainly with a migration program, and we started to now wonder, is this blebbing driving potentially cell survival, which is really at the core of what a metastatic cell needs to do. And so we are faced again with the same challenge. We somehow need to be able to manipulate morphology, and through the manipulation of morphology, we potentially could manipulate cell survival. Well, we were lucky here in this particular case. What people in the lab figured out is that when they take beta-agglutinin, short WGA, and begin to dope cells with this reagent, then they can decrease in a dose-response fashion, decrease the fraction of the surface that actually forms these blebs. And so Andrew Weems took now that trick and applied that to an MV3 melanoma cell, where the adherent form of these MV3 cells really doesn't change much in its behavior as the WGA concentration goes up. However, the detached population of cells begins to have, compared to untreated cells, begins to have more and more cell death. So in other words, bleb reduction leads to increased cell deaths, which would support the hypothesis that blebs are actually a survival organelle. And so he repeated that in a variety of cells. M498 is another melanoma line. That melanoma line is a little less adherent to begin with, and therefore is responding more to this WGA treatment. And then he went into the A375s, and the A375s have absolutely no response, whether adherent or detached. And that was a peculiarity, and I will use that to explain some of the things I'm going to show, so keep that in mind. But when he then, he had again a serendipitous observation that when he cultured these cells to full confluency so that these cells became quiescent or pre-stressed, then applying WGA to the detached form again leads to increased cell death. And when he challenges these cells with our MAP kinase inhibitor cocktail, the same behavior, right? So that means that these plebs might be very similar to the Lamalipodia story I showed you, might be actually a defense mechanism for cells to survive under growth challenged conditions. I don't want to belabor that point too much. Manipulating morphology, again, is a tricky business. Are those just wholesale effects? And so Andrew developed a few other assays in which he can, again, perturb pleb formation, as shown here, for example, with the hydrogel, and we see exactly the same behavior in terms of increased cell death under pleb disruption, pleb formation disruption. So that brought us really to the questions, are dynamic plebs promoting survival signals? And again, to go from morphology to an intracellular signal is a rather tricky step and needs a lot of luck. Well, we had the luck again. What Andrew observed is that cells which pleb accumulate septin puncta. And so you see that very nicely in this, again, Z-scan here, where in between the plebs, you have these bright fluorescent signals from the septins. So what are these septins? Well, these septins like to go to curvatures of about 1 micron radius. And as this three-dimensional analysis here of the curvature of this cell shows very nicely, is that exactly between plebs, you tend to have negative curvatures of about 1 to 1.5 microns, which is exactly conducive to the assembly of these septin scaffolds. And so we can test that now in another way and directly show that when we abrogate, again, pleb formation, like with the WGA or these hydrogels or a few other ways, then these septin puncta actually go away. They not go away totally in 3D projections, but when you go through the individual slices, you really see that these puncta between plebs here are wiped away. And when we now go and compare again the cell conditions that were responsive to pleb inhibition versus non-responsive to pleb inhibition, then you see very nicely that the A375s, which were non-responsive to pleb inhibition, have, even under no WGA treatment, have no septins at the cell surface, while all the other cells have these septin puncta. So the question then is are these septins really promoting survival signals? And so what Andrew did then is a bioID proximity proteomics assay, and very peculiarly found that NRAS, and in the MV3 case, it's actually a mutated, constitutively active NRAS in this melanoma, that NRAS is in high proximity to septins. And when he biochemically pulls down septins, pulls down NRAS with septins in septin positive cells like the MV3 or the stressed A375 forms, then NRAS comes down, while again in the unstressed A375, which doesn't have septin puncta, NRAS doesn't come down. So these are pretty convincing proofs already that septins might really pre-organize this constitutively active NRAS form. We can take our imaging assays and our computational assays and really quantify beautifully now that NRAS and septin puncta are very, very highly co-localized, right? Versus as a control, septin versus collagen as a background signal has no correlation whatsoever. So this seems to be very specific, right? And so with that in hand, Andrew could now take, of course, septin inhibitors, and there are septin inhibitors like FCF, or poison septin through mutations, and test again directly whether the disruption of septin puncta would now lead to increased cell death, very much like direct perturbation of plebs has. And I don't want to belabor that data. You see it's smack on. It's exactly that prediction is supported. So you need these septin puncta in place to have these detached melanoma cell lines survive. And then if you take the septin puncta away, they begin to die, right? So what is downstream of RAS? To this audience, I don't need to explain that deeply. PI3 kinase is just there. And you see very nicely when we now go in and perturb, or Andrew goes in and perturbs septins or BLEBs directly, then he reduces actually PI3 kinase activation as measured by imaging. And he can do the same thing for MAP kinase activation, again through a biosensor, and has shown that blebbing is essential for upregulated ERK signaling in these cells. Just to prove even more directly that it is really NRAS mediated, we express now in these MV3 cells, excuse me, a dominant negative form of NRAS, and show again that with taking away or competing against the constitutively active NRAS mutation, oncogenic mutation, we can actually push that cell death up. So that leads us to a model in which we have, in this case, really adhesion loss leading to BLEBs through detachment, but there's many other environmental conditions that lead also to blebbing. These BLEBs lead to aggregation of septin scaffolds. These septin scaffolds organize NRAS and PI3 kinase and also ERK signaling downstream, and that leaves us then with a survival signal. So a second example where a morphological program like blebbing is directly responsible for something we really look for when we study oncogenic cell responses. And in this particular case, the oncogenic response would be cheaply understood as the result of constitutively active mutation of an oncogenic protein. But that alone, and I really want to emphasize that, that alone does not work, because all these experiments we have shown you, which require morphology, have this constitutively active NRAS expressed, and that alone is not enough to drive the survival signal. And so that brings us to a very interesting question. Why does a constitutively active signal need spatial organization to be oncogenically penetrant, right? One possible answer, and we don't know that yet, is it the scaffolding which enhances effector interactions? Or is it the proximity to BLEBs, which now might upregulate through this diffusion trap I showed at the beginning of my talk, might upregulate now non-linearities in signaling that now pump up these pro-proliferation or pro-survival responses of these cells, right? And then the second question, of course, if this is all sorted out, is there potentially an optimal configuration for constitutively active signals to be oncogenically penetrant? So, for example, does cell polarity help? By aligning these scaffolds on one side of the cell, you might have, again, nonlinear upregulation of these oncogenic signals, and there's many more things. And so the lab is working now really mathematically at getting at some of these questions. I would like to end this part really with what Andrew calls the Hail Mary experiment, right? We were curious about normal non-transformed cells and this Anoikis response when they cannot reattach, right? Which that movie already showed. And so when Andrew goes in and looks 2 hours after detachment at a fibroblast, well, they have, again, no septins or only very weak septins at the cortex. However, if he films these cells 30 minutes after detachment, so long before they stop blebbing, as long as they can still bleb, they have septin punctum. And so the question is, of course, can we potentially now maintain dynamic blebbing and through that elevate Anoikis resistance also in an untransformed cell? And so again, this is a huge challenge, right? And again, through a lot of luck and ingenuity and very, very good hands, Andrew was able, and this is fresh off the press, was able to do the following experiment. And as I already alluded to, these cells probably stop blebbing after 2 hours because endocytosis chews away the plasma membrane. And so what Andrew tried is now to take a very mild mutation in dynamin, which is an essential component of the endocytic machinery, and express that mutation in normal fibroblasts so that maybe they still maintain membrane a little longer. And that's exactly what he achieved. You see all these cells now begin to bleb. So this is at the same time point filmed as the normal fibroblasts without mutation. And see what happens here. What happens is that the control condition, as we all know, in detachment, they die. But when you now block or reduce endocytosis, you come down here to the green dot, so you die less. And when he now takes these cells which carry that dynamic mutation and treats them again with bleb inhibition or septin inhibition, they actually rescue again the control condition. So this is directly demonstrating that this morphological program of blebbing is also an effective pro-survival factor in normal cell if normal cells could bleb indefinitely as cancer cells do. So this brings me to the summary of story 2. Blebs in this specific case lead to NRAS and PI3 kinase, and I forgot to mention here also ERK activation, which is obviously in this case a pro-survival signal in cancer cells. And we had a third story. Some of you might be familiar with that story, also in metabolism, where stress fiber formation, very much in the same spirit as morphological control through lamellipodium plebs, lead to increased glycolysis, which then also again leads in cancer cells to increased pro-survival and pro-proliferation responses. So shape really matters. I'd like to spend the last 2 minutes with really where does this go next, right? These are beautiful cell biological experiments which we can now do thanks to our 3D imaging. But it is all still ex vivo, right? And we are now at this point where, like 8 years ago, we went in full speed to develop the technology to look at molecular processes ex vivo in 3D with no resolution reduction. We want to do the same thing now, really in the mouse as a next level of experimental system to really go after these tumor and metastases signaling effects. And this is really work done by Kevin Dean, who is an assistant professor in the department, and his team, his still small team, Hazel Borges and Chin Long Lin, who worked out that story together with Andrew Weems and a couple other postdocs in my lab. So let's look whether these morphological programs happen really in primary tumors. I'm not going to belabor here the technology. Again, it takes a lot of physics and a lot of engineering and a lot of computer science to get at these things. But what you saw here is now really a primary tumor of a melanoma inside a mouse. And we have pretty much the same 300 to 400 nanometer resolution to look at subcellular effects inside the primary tumors. And when Andrew now looked with this instrument at his particular melanoma, which again expressed septin 6 GFP, then when he goes further back into the tumor, where cells are probably less in an amoeboid phenotype, so less blebby, then he doesn't really see septin puncta. But now with this resolution, this enormous resolution these microscopes deliver, when he goes at the front of an invasive tumor, then these septin puncta begin to pop up. It's still a little hard to see the blebs, and you bet we are working very vigorously on that to really show the full program morphology to septins to signaling now inside a mouse, right? But still sort of hand selected, and this is a little bit cheating, right? Andrew shows you here the same, pretty much the same septin signal type inside a tumor stroma of individual cells he found there, as he sees it just in collagen. So that shows that the stories I told you today are potentially actually really happening in vivo. And this is a last slide really to invite everyone. We just got a NCI grant funded to build a center that pushes microscopy and computer science to do in situ imaging of metastatic tumor formation, but can also be primary tumor formation. And so we would really love, maybe starting in a year, to collaborate with the community, not in Dallas, who is interested in using these tools. And with that, I would like to go to my last slide. It takes a large team of green people at the bench, of yellow people at the math desk programming to really pull this off. And I think I acknowledged each individual, but it is really 20 people working together and thinking together to come up with the results I was able to present on their behalf. Thank you very much.
We can virtually applaud Gowden's. That was really terrific. So we have a few questions here. First is from Joan. I'm guessing this is Joan Frugi. And she asks, have you looked at the role of CDC42 in the blebs? We saw that blebs associated with mutant p53 and suspension were dependent on CDC42, and CDC42 is known to control septin rings during cell division, certainly true in yeast. I forgot to mention also that CDC42 has been shown to be required for RAS-driven anchorage independence.
Right. So we haven't looked at CDC42 specifically. We certainly would have the capabilities through biosensors for CDC42 to do that. However, I'm actually glad that this is brought up. We have another story cooking, which I had absolutely no time to show, where we looked a little bit deeper at what happens really in blebs and why are blebs actually polarized. When you carefully look at these cells, these blebs are not all around the cells, but they are in particular places. And the story there points very much like into a RAC CDC42 activation in the bleb. And of course, this is peculiar because people think that blebs form through pressure and not through regulated signaling upstream of the cytoskeleton. And we actually have some evidence, so does Mathieu Peel in Paris, that actually bleb formation is probably much earlier than the current literature suggests, really dependent on actin assembly downstream of, in our case, it's RAC, so we sorted out the RAC pathway, but I wouldn't be surprised at all if CDC42 goes in parallel. So while for this particular story, we have not looked into CDC42, I fully believe that CDC42 has a huge function in bleb formation and maintenance.
Excellent. Here's a question from Greg Bernet, who says, amazing talk. Regarding the autoencoder, could you figure out what the pattern feature is activating feature 57, the most discriminating feature in your encoder?
Yes, we are. And so I refer really to the paper for the technical details. There is the paper and a very, very long supplement, as always in these rather provocative technology development stories. We measure the latent feature power really through the correlation that we find to the classifier score, right? And so I showed you the simplest case scenario where we have one number really standing out by a factor 3 or 4. So it's not a full order of magnitude where this number is stronger than anything else. But still, it is very, very highly significant statistically. But in other autoencoder trainings, you will not have a single feature, but you might have a combination of features, right? And so that example I showed where we move now just along feature 56 to make a non-metastatic or low metastatic cell highly metastatic in terms of image generation, what you need to do now in the latent space is just walk along a curve of the most dominant features. So that's potentially a non-linear trajectory you need to walk through, and this is all demonstrated in the paper. For this audience, I just didn't want to go into these mathematical technicalities.
So there's no specific biological thing that you can say is what is measured by?
Okay, sorry, maybe I misunderstood the question. Right? Okay, so that's of course the power of this analysis, right? So what this autoencoder learns is you would never come up with this particular image phenotype I showed you. And again, the reason we were able to see that in the end is because we exaggerate, right? We extrapolate the latent space, right? So we would never make that discovery in the direct image. And so what the autoencoder learns is that potentially a combination, a highly convoluted combination of image properties that drive now that functional classifier that comes out on the other end, right? And so this is a very generic algorithm we developed here, and actually Google now just last month published something similar, where they can now convert cats into dogs with exactly the same idea, right? So you can download from Google software which does that. So this is the idea that you learn driving features in a completely unbiased fashion. And as a human, you have no way of predicting these features upfront. That's the key message.
Great. Actually, if I may, I have a couple questions. My first question is about the impact of dynamic blebbing on growth factor receptor, plasma membrane occupancy, and endocytosis. Do you know if we're altering the functional population of growth factor receptors on the cell surface?
We have not looked into this at all. I'm not so familiar with the bleb literature that for us the bleb is a tool to look into cancer. So I couldn't cite now a paper which looked at that. That doesn't mean that it doesn't exist, right? But these are also very, very, as you can imagine, David, very tough imaging experiments, right? Going into
Sure.
imaging receptors on bleb specifically, right? Yeah.
I would guess whether that endocytosis, would you expect it to be suppressed in the blebs?
Again, I don't know of someone who has tried to image endocytosis specifically in the bleb. There is a very well-known link between blebbing and macropinocytosis, but that happens again at the bleb next, right? So that's well described.
Yeah, and then this is probably like biologically infeasible, but I'll ask it anyway. I wondered if there's any way to ectopically trigger septin-6 assembly to try to deconvolve the septin assembly and the morphological changes. Would it actually be feasible to concentrate them like on the cytoplasmic surface of a mitochondria and test whether that launches RAS signaling?
Right. So that's a very clever proposal, and you bet Andrew Weems is deeply thinking about that, and he's on the job market next fall. So that's certainly an experiment he would love to do in his own lab, right? So we are not gonna try to do that.
Okay, it looks like we have no more questions, so let's just virtually applaud one more time and thank Gavin for a terrific talk. Thank you.
Thanks everyone who listened, and thanks again, David, for the invitation. It was a great pleasure and honor to speak at the Farber.
Next time in person.
Absolutely. Stay well, everyone, and enjoy the rest of your day. Bye-bye.